Let's continue our discussion of biological molecules with proteins. So proteins are really important in living cells. They perform a lot, well, really all of the functions in a cell, and are really sort of the worker bees. I like to call them that, of the cell. They perform all of the functions. They provide a lot of structure. So they really are a very important biological molecule. Proteins come from our diet. They're found in meat. They're found in dairy products. Some plants, such as soy, contain protein. And as I said, almost every function of a living being directly depends on a protein. And we'll talk about some of the types of proteins and some of their very specific jobs in just a few minutes. Proteins make up a huge percentage of a cell. So we talked about lipids, phospholipids specifically, forming the membrane around the outside of the cell. We have our carbohydrates that are providing energy storage. Our lipids are also providing energy. They're also our precursors to our hormone molecules. And then, of course, we have our nucleic acids that are the genetic material. But proteins are the most abundant type of biological molecule in a cell. And proteins are a little bit different from the other biological molecules in that they can have incredibly complex structures, very diverse structures, very complex structures. You can have chains of amino acids, which are building blocks for proteins, sort of folded and twisted and into a variety of different structures. And that means that proteins can take on this diverse amount of function within a cell. And proteins were actually thought to originally be the genetic material because they were so diverse, at least the genetic material that was storing and coding heritable information. So proteins are polymers of amino acids, and those amino acids form an unbranched chain. So they are a chain of monomers, just like our carbohydrates and like we'll see in our DNA. But one of the defining characteristics of protein is that it has multiple layers of structure. So the first layer of structure is this unbranched chain of amino acids. Unlike carbohydrates, where you tend to have sort of the same monomer repeating or a very small number of monomer types in a complex carbohydrate, there are actually 20 amino acids that can be repeated in any combination and variety of sequence. And those amino acids have a basic structure, and each one is characterized, again, by the functional groups that are attached to it. So amino acids have a basic structure and some general characteristics, and then they have these functional groups called R groups that stick off of them that give them more specific characteristics within the protein. So all of our amino acids have this same structure represented in purple here. There is an amine group, that nitrogen group there, that's where it gets its name, 2 carbons, and then that carboxyl group, the carbon-oxygen double bond here. That's where we get our acid. So amine for the nitrogen and acid for this group here. That will release a hydrogen. So remember, our acids release hydrogens into a solution. And then we have these different R groups attached. So the R groups have some characteristics. Basically, they fall into the same types of molecules that we've already seen. We have our non-polar amino acids. Those are the ones that are not going to interact well with water and are therefore hydrophobic. We have some polar groups that are therefore hydrophilic and interact well with water. We have also acidic and basic amino groups. So those are groups that are going to donate hydrogen, or hydroxyl groups, or pick up hydrogen from a solution, and those tend to be charged. So the acidic groups tend to be negatively charged and the basic groups tend to be positively charged. And again, they all affect the structure of this protein and how the protein will fold on itself and produce its overall structure, and then also the function of a protein. And the functions of proteins are very complicated, and we'll talk about some of those in a minute. But just keep in mind that amino acids have the same basic structure, but these differentiated R groups give them very specific functions within proteins. So because we're talking about polymers, we need to string these amino acids together. The polymer that is produced from a string of amino acids is called a polypeptide. Poly meaning many. Peptide is the type of bond that forms between an amino acid. So again, this is another bond name that we need to remember and recognize. Our carboxyl group on our amino acid, on the C end, the C terminus of the amino acid, is linked by a covalent bond called a peptide bond to the N terminus, or the amino end of the next one. So again, we have our repeating units in a very specific direction. And just like our carbohydrates and our lipids, also linked by a dehydration reaction where water is removed from the two amino acids and released in the formation of those peptide bonds. So here we have a really simple polypeptide. We have our amino acid ends and then our R groups sticking off of that amino acid backbone. So the really important thing about proteins is that they make this really diverse structure. And that linkage, that polypeptide linkage of those amino acids is really just the basis for protein structure. In order to make a functional protein, that polypeptide has to be folded and processed. So the actual outcome of that dehydration reaction to produce a polypeptide is just a chain of amino acids. And when we get to the process of translation and the production of amino acids, the process of translation actually produces a polypeptide, not a protein. The protein is not functional until the cell modifies it and folds it into very specific structure. So this is an example here of a very specific protein structure. We have an antibody protein. And if you look at the center of this picture here, we have the antibody protein shaped to recognize a very specific protein from a flu virus. And so there we have our structure and function again. You see the shape of that antibody protein reflects the shape of the protein, and this allows the immune system to recognize that protein from that flu virus and to initiate a response to break up or to attack that flu virus. And that function would not be available if it weren't for the very specific folding that happens when those proteins are produced. So protein structure comes from several layers of superimposed structure of proteins. All proteins have three levels of structure, and then some have a fourth. So let's talk about those different levels of structure in proteins. We already sort of saw the primary structure. That's simply the amino acid chain linked by those peptide bonds. So this is just a polypeptide. It's just monomers of amino acids linked together to produce this polymer, this polypeptide. At the top we can see our amino acids represented in purple, and then the R groups coming off of them. If you look at the top, we see that the R groups are alternating. So the R group is at the bottom, and then the top, and then the bottom, but we still have that N terminus end, and then we have the C terminus end. So proteins have a direction. One end has a free amino group, the other end has a free carboxyl group. So amino acids are added always at the carboxyl end. So primary structure is just the linear sequence of amino acids. After that, that linear sequence begins to be folded into what we call the secondary structure. So there are two types of structure that characterize secondary structure, and they are both produced by hydrogen bonding between R groups. The first I'm going to talk about is called the alpha helix. So you can see it's sort of this ribbon, this twisted ribbon, to make a really nice sort of twisted structure, almost like when you curl a ribbon on a package. So we have our, we see the carbon and nitrogen backbone there, and then the R groups sticking off. And it is the interaction, the hydrogen bonding interaction between those R groups that holds this alpha helix together. And so these are going to be our, generally our polar R groups arranged in a particular fashion so that they, at a repeating distance, they interact and produce these hydrogen bonds to make this ribbon structure. Our second type of secondary structure are the beta pleated sheets. So beta pleated sheet sort of looks like an accordion. Each of these amino acids sort of form this up and down pleated structure, and they interact with the pleated structure next to it, forming hydrogen bonds between those R groups. So if we were going to continue this beta pleated sheet, we would actually see the first strand of that beta pleated sheet, and it would sort of bend around and produce a second sheet. Sorry, I clicked that a little bit too soon, but this image actually shows that a little bit better. So this is spider silk protein. Spider silk is really, really strong. Think about that for a minute. Remember, the beta pleated sheets are held together, and also the alpha helix are held together by hydrogen bonding. And remember, hydrogen bonds are collectively very strong. So spider silk proteins are a collection of these beta pleated sheets. And you can see down here we have our beta pleated sheet, and then it turns, and our next sheet turns, and our next sheet, and so on. The reason that the spider silk is so strong is that you have the hydrogen bondings in between, but you also have the ability of this protein to stretch. So they can stretch without breaking, and that really is what gives spider silk its unique strength. And you can see in that picture in the middle that you have these repeated regions of beta pleated sheets. And so when you have proteins with beta pleated sheets, you can sort of have this stretching, but you also get a lot of strength within it. Our next level of protein structure is called tertiary structure. Before I talk about what tertiary structure actually is, I want to show you that in this molecule here, we have an alpha helix, we have some beta pleated sheets represented by these straight line ribbons, and then the tertiary structure is the way that those secondary structures are held together. So in a single protein, you can have regions of alpha helices, beta pleated sheets, and even regions where you really just have your chain of amino acids, but those regions are folded together and situated together to give them a specific structure. So tertiary protein structure is held together by interactions between R groups, but they can be sort of any chemical interaction that you can think of. You can have hydrogen bonding holding together these different regions of folding. You can also have van der Waals interactions or other hydrophobic interactions. Those are sort of similar to hydrogen bonds in that they're like temporary interactions or attractions between two molecules. You can also have covalent bonds, so that is shown there, that disulfide bridge there is a covalent bond, a strong, almost permanent bond that you're going to get between those two regions. And then you can have ionic bonds. Remember, ionic bonds are attractions between positive and negative. And so you get this interaction with all of these different types of bonds here holding together that overall protein structure. So all of our proteins are going to have up through tertiary structure. It doesn't matter what protein you have, you're going to have at least tertiary structure. Some proteins also have a fourth level of structure. So quaternary structure is the interaction of multiple tertiary structure proteins together. In this example here, we see a protein called transthyretin. This is a transport protein that transports a thyroid protein to retinol. And it's just a really good example of quaternary structure. So we see there that we have four subunits there, kind of two light purple, two dark purple. These are four identical polypeptides folded into tertiary structure. And when they're put together, they produce this functional transthyretin that moves through the bloodstream. We also have collagen down there. Collagen is a triple alpha helix protein. So each of the individual polypeptides is a alpha helical ribbon that is twisted together to produce collagen. So collagen is a structural molecule. And that's why it sort of looks like a rope there. It's a really strong molecule due to the interaction of those three polypeptide units. Sickle cell is an example that we are going to talk about quite often in this book. And it's a good example to talk about here because sickle cell is a disorder that results from a change in the primary structure of the hemoglobin that affects the tertiary structure of the molecule. So sickle cell anemia is a disorder where the red blood cells don't form properly, and instead of having those nice round plump disc-shaped blood cells, you get this sort of crescent moon shaped red blood cell. And when that happens, the red blood cell cannot bind and transport oxygen the way that it should. So this diagram shows the correct primary structure of a normal red blood cell. And with individuals that have sickle cell, they have a change in that sixth amino acid there from a glutamine to a valine. When that happens, that affects one of the subunits, one of the tertiary structures of that molecule. And so when they're put together to form their quaternary structure, instead of producing a molecule that effectively transports protein, it can't do that. If we look at sickle cell, we have that one subunit is misshapen, which produces hemoglobin molecules that want to stick together. And so the proteins stick together or aggregate together, reducing oxygen carrying capacity. This is a problem not only because there's reduced oxygen transport to muscles in the bloodstream, but also because those abnormal hemoglobins can actually clog up small passageways and things and block proper blood flow. And so you can have problems with blood flow and oxygen transport, and also a lot of pain if these get sort of stopped up in the organs. So now that we've talked about the structures of proteins, the overall structures, let's talk a little bit about the types of proteins. There are 8 types of proteins, the most important of which I would say are enzymes. So enzymes are proteins that sort of mediate chemical reactions. If you're familiar with the word catalyst, a catalyst is a molecule that increases the rate of a chemical reaction. So an enzyme is a molecule that increases the rate of metabolic reactions, so reactions that happen within living organisms. So this might be a protein that works in the chain of events that breaks down food and converts it into stored energy. It might be an enzyme that helps with the production of a specific amino acid within the body. Enzymes are going to mediate all of those processes, so they keep our metabolic processes moving correctly. I'm not going to go through each of these different categories of proteins. I would like you guys to be familiar with the different types. So we have storage proteins. So this is proteins that store amino acids, hormonal proteins. So these are proteins that sort of regulate the processes, the hormonal processes within our body. And then we have some other ones. So if you guys would just read through these, I would just basically be reading them to you just to be familiar with what they are. We will come back to a lot of these later, either in 101 or in 102. I got a little bit ahead of myself. I already talked about enzymes, so I'm not going to repeat that. But this is just a little summary of what enzymes do. So we've talked about protein structure, and we've talked about structure and function, and how the structure of proteins is very tightly fixed with its function. So we saw that in the red blood cells. We saw that in our example of quaternary protein with the collagen. So if that structure gets disrupted, the protein becomes dysfunctional. So protein structure depends on that original amino acid chain sequence, but the physical and chemical environment can also affect protein structure. And so there are a variety of physical and chemical situations that will cause a protein to break apart or to become dysfunctional. When that happens, it is said to be denatured. So this cycle here shows a normal protein. Under certain processes, the bonds between the secondary and tertiary structures can actually be interrupted, and that protein can become dysfunctional. This might be temporary, it might be permanent, it depends on the protein, it depends on the situation. Some of the physical, environmental situations that might affect these are like heating up. If a system gets too hot, the proteins can become denatured. Chemicals, if a cell becomes too acidic or too basic, you can disrupt the structure of the protein. So cells have to maintain the proper environment for their protein structures to be functional. So our last category of biological molecules are the nucleic acids. I'm only going to talk really briefly about nucleic acids because when we come back to the structure of DNA and DNA replication in Chapter 13, we always have to review anyway. So basically for our nucleic acids, I want you to sort of know the general features of it, but you don't need to know as much detail right now as our other three. So the monomers that make up our nucleic acids are called nucleotides. And nucleotides produce both DNA and RNA. We'll talk a little bit more detail about the differences later. They are linked by something called a phosphodiester bond. So here we have a nucleotide, we have a sugar, our nitrogenous base, and our phosphate group. These are the basic monomers of either of our nucleic acids. And they are linked between a hydroxyl group on one of the carbon rings to the phosphate group on another nucleotide. So again, we have a dehydration reaction where we lose water. It's written here as condensation reaction. That's just a synonym for dehydration reaction. And in this process, you produce what we call a phosphodiester bond. So remember we saw the ester bond in the fats, and that is a link between a carbon and an oxygen with the phosphate in the middle. So here we have the phospho, and then the two oxygen is the diester bond. So you do need to know the name of the bond that links these nucleotides to form our nucleic acids. So the monomers of our nucleic acids, whether they're DNA or RNA, are the nucleotides. So here's a better picture that just shows the individual pieces. We've got our phosphate group coming off of a 5-carbon or a pentose sugar ring. And then we have our nitrogenous base. So the nitrogenous base can change depending on the nucleotide. There are 4 nitrogenous bases in DNA and 4 in RNA. And the 2 that differ are the thymine and the uracil. So DNA contains thymine, which is a more stable base than uracil, which is found in RNA. And we'll come back to that a little bit later and talk about why we have some of these differences between DNA and RNA and what other differences we have. But when we say we have a DNA sequence for something, we mean we have figured out that sequence of nitrogenous bases. The phosphate and the sugar stay the same. They produce what we call a backbone for this molecule, and then the nitrogenous bases change. And that's what we are talking about when we say we know the sequence. Sort of the general shape of DNA is what we call a double helix. So there's a lot going on in this picture. If we start at the left-hand side, we see the sugar and phosphate groups with the nitrogenous bases sticking off. And those sugar phosphates form what we call a sugar phosphate backbone, which is a really stable covalent alternating sugar and phosphate chain, with the nitrogenous bases coming off of it. Those nitrogenous bases will specifically bond to nitrogenous bases off of a second sugar phosphate backbone. So these blue ribbons represent one sugar phosphate backbone, and then the second represents the other, with the nitrogenous bases attracted in the middle by hydrogen bonding. So the nucleotides are linked by covalent bonds, those phosphodiester bonds, and then 2 strands of DNA are linked in the middle by hydrogen bonding. RNA has a slightly different shape, and the shape depends on the specific nucleic acid that you're talking about. RNA is a single backbone that is folded into a very specific structure. And again, we'll come back to those RNA molecules. There's a variety of them, and they are all single-stranded with a very specific structure because they have a very specific function. So this is really all we're going to talk about for our nucleic acids, just to sort of give you a general idea. And we'll talk about some more specific characteristics and how those relate to the process of transmitting genes and using DNA as instructions. But we'll come back to that a little bit later. Just sort of an overview of this process of going from DNA to what we call the phenotype, or the physical expression of that information, is this process of gene expression. So going from the coded information in DNA all the way out to something functional in an individual. So during this process of gene expression, we go from the double-stranded DNA through a process called transcription, where we produce a molecule called messenger RNA. So messenger RNA is a single-stranded RNA that's going to code for that amino acid polypeptide. And that process of going from the mRNA to the amino acids is called translation. And then once we get that amino acid chain, of course, we need to fold it into our functional protein. So the protein that we have here is called the crystalline protein. It is encoded by the crystalline gene, and that crystalline protein is found in the lens cells in our eyes. They're a protein that actually absorbs some of that light and is then processed so that we can see a particular image. So this sort of concludes our discussion of biological molecules. Remember, we had 4: carbohydrates, lipids, proteins, and DNA. You do need to know the monomers of each of them. You need to know the type of bond that forms. You need to understand dehydration reactions. And you also need to know the major functions of each of these molecules within the cell.